明交大生科院微民所许博琛助理教授演讲。
院长好，然后各位同事大家还有各位同学大家好。那这应该是我们本学期的第1次周3学，然后很高兴我作为院所所长陈彦荣，然后很高兴跟各位介绍我们所上看了好久，终于费尽千辛万苦，终于找到的新老师，我们最年轻的许博琛老师。那我负责为大家介绍一下许博琛老师的背景。那许老师其实是一个标准的大人，然后从大学部，然后硕士与博士都在清大，然后那清大毕业之后呢，许老师就到中研院军医实验室做博士后研究，然后博士后研究做得很好，然后许老师就把他留下来做 Senior Science。然后，所以，那我们当初在征人的时候，我们就发现，吕老师对于许老师的这个推荐非常的坚强，然后而且，许老师已经在许老师实验室已经建立了一些，已经发表了一些corresponding 的 paper， 它有非常好的 dependency， 所以我们就向学校推荐许老师来担任我们的新的这次 professor。那许老师的题目，他是用 Easter 用这个酵母菌做模型，然后研究这个细胞演化的分子的机制。然后另外他也是一个 Cultivating 的专家。然后所以我们把剩下的时间交给许老师，让他为各位介绍他的研究。
It's really a great honor to share with you my current research. So the main topic today actually is I just try to introduce how I can use multiple yeast species as a tool to study the evolution of some molecular system. For example, the transcription regulatory network system. Okay, so basically it looks like it's the evolution talk, but actually I'm still a molecular biologist. So the main topic I will focus on 3 parts. Basically is using to study to characterize microevolution of transcription regulatory network in diverse yeast species. So I will compare yeast species and their transcription regulatory network. And also I will use yeast as a model to study the microevolution for the perturb the transcription regulatory network. And then I will use the experimental evolution to study the organelle evolution, basically it's mitochondria in yeast. How can we use yeast to answer this kind of question and use this as a tool? Okay? So basically, before getting into the real project, so I want to just using one slide to simply remind you the current fundamentals of modern evolution biology. So basically, you need to know what is evolution. Evolution is actually the species that change over time, and in the end, to accumulate enough change and allow the species to become a different species is what we call speciation. And this lineage from one species to another species, actually, this is like with a clear track. So we call this, they can share a common ancestor. It's a very ancestral lineage we can observe. And then, because the modern evolution biology, including DNA, we know everything starts from DNA. So you know that actually the evolution change, what we mean, initiate from mutation in the DNA, in the genotype, and then lead to the phenotype. So you know this, and then we know that how species can evolve. There are 3 important factors. One, they have to have mutation to allow them to have adaptive innovations in anything, any phenotype related to their survival. So which means they have adaptive innovation in the survival strategy to allow these species with different changes can maintain after a long period of evolutionary divergent time. So once you know this, change, mutation, adaptive innovation, and the natural selection, you know the basic of evolution. So that's one slide can help you. You notice you are expert of evolution biology right now, okay? And then we know that from genes to cells, organs, and population species, and different clade, this is increase the complexity of organism. So we can simply define evolution into two big categories. One is microevolution. Basically, it's the difference below the population level, below the individual level. For example, polymorphism in the DNA, in gene, we call this microevolution. And basically, they just give a small change. For example, a bacteria develop a new drug resistance from probably getting several mutations in some specific gene, drug-resistant gene. We call this kind of change microevolution. But if the evolution change is above or at species level, for example, we compare chimpanzee with human, we compare fish with bird, you know they are very, very different. So we call this big difference, we call macroevolution. So under this, understand this concept. So this is the favorite things our evolution biologists like. We like to build evogram. What is evogram? Actually, it's a kind of diagram that showing the trajectory of specific feature or specific trait evolve. How this trait evolve from a group of organism or a group of organisms. So you can see, for example, the evolution of human actually evolved from a lot of different key evolution stage. From like, we assume that the life starts from a prototype of cell. So they develop into multicellular life. They develop to animal life with, they can move, they have digestive system. And then they become like, develop the spinal cord, and then from the aquatic environment go to the, try to survive on the land, and then they become smarter and started more like the Homo sapien in the current species. So develop this kind of, to build this kind of hypothetical evogram is the dream of evolution biologists. So this idea can be applied to study evolution of anything, like my case, transcription regulatory network, okay? So, but to understand the evogram, okay, evolution trajectory, we need to know a lot of things. This is just an example, okay? For example, like what we have right now is only fossil record and the current species or extant species. So if we want to build the difference between some kind of ancestral species, we will never see it, right? So that's why we can only base on the current information and probably some information from the fossil record in order to hypothesize how a feature, what kind of organism evolved from their hypothetical ancestor to the current species. Okay, so similar idea we can apply to microorganisms like yeast and or molecular system using yeast. And the issue is that a long time ago we can only observe the feature from fossil record and extant species. But now, right now, we know DNA, right? We have genome sequencing project, we know DNA. So that's why we can easily use the DNA to build the phylogenetic tree as long as we have enough genome from current species. Yeast is an example. We have tens of thousands of species. Of course, like, doesn't mean everybody have the proper genome, but the current genome is already available enough for us to do a lot of analysis. So we can use the DNA sequence to be of our interest gene or conserved gene to build the phylogenetic relationship between different species, right? So then we can focus on a specific feature and then know different type of feature in different species. And based on this phylogenetic tree, what we can see is only the current. What we can see is only the current species, right? But based on the parsimonious strategy, so we assume that the early divergent species is probably the one that is more close to the common ancestor. So based on the divergent time from earlier to late, so we can simply assume using parsimonious idea that this kind of feature is evolved from D type to C type and then to AB type. And based on the similarity of this, we will assume probably A type is evolved from B type, okay, because of the similarity from here to here. And also we can also hypothesize that probably D type in the current species is hypothetically more close to the real common ancestor, but we will never know. Okay. So what is parsimonious idea? So it's that without further evidence, we will assume that the evolution change from one type to type, we are using the most simple and straightforward way. So which means evolution from D type to C type is direct change. We are not going to assume that they are going to all have this change first and have another change, then lose this change and become this. So this is not going to happen. So using a parsimonious idea, we will assume D to C is direct change. So using this strategy, we can try to hypothesize or to build the evolution trajectory of anything we want to study. Okay? So go back to my research interest one. Actually, I will use my published paper to describe my research interest. So I focus on the transcript regulatory system in the microevolutionary pattern. So assume that we have two current species. Usually we will observe that the orthologous transcription factor will have different regulated targeting. So this kind of pattern difference between different species, for example, two yeast species, is a kind of macroevolution we want to observe, we want to characterize, and want to study. Okay? So we give this microevolution in a transcription regulator network a term called transcription rewiring. So basically, you assume that two species start from a common ancestor, and then they have the orthologous regular, which means target gene, group of target gene, will regulate specific genotype. But in species A, transcription factor X regulate this gene, right? But in species B, another transcription factor regulates the same set of gene. And the orthologous transcription factor X in species B change to regulate different target gene and probably responsible different phenotype. So you can see between species, there is a TF substitution between X and Y, and between the same regulant, they have a regulant handover for X and X, regulant 1, regulant 2. So this kind of TF substitution and the regulant handover is what we call transcription rewiring, and it's a specific pattern of macroevolution. And we use yeast to study it. And give you a true example. So in Candida albicans is a pathogenic yeast, Saccharomyces cerevisiae is bakery yeast. So they diverged from 300 million years ago. And you can see that there's a specific ion responsive gene we call ion regulase regulated by SAF1 transcription factor in Candida albicans. But in Saccharomyces cerevisiae, there's another set of FT factor regulated gene. And the orthologous SAF1 or orthologous FT factor in these two species regulate different things. Okay, so this is a typical case. So we know that this transcription that belongs to ion responsive transcription regulatory network. So we expand this idea because we focus on all of the current available ion responsive regulatory system in fungi. Okay, so currently there are 3 types. Type 1 from this species. There are using two repressors to regulate the ion uptake gene and ion utilization gene. So under the ion replete condition, repressor will shut down the ion utilization gene. Under ion replete condition, the repressor will shut down the ion uptake gene. And the type 2, there's one more activator get added into this system. So this activator will be responsible for the induction of ion uptake gene under the ion depleting condition. But there's another 3 types completely different. This happened in Saccharomyces cerevisiae. They use a different activator and a post-transcriptional regulator to regulate ion uptake gene and ion utilization gene. So these are 3 types. But if we put all of the ortholog of this ion regulatory gene, regulator transcription factor together, this is a post-transcription messenger RNA binding protein. So 4 transcription factor and 1 messenger RNA binding protein across the biological tree from Saccharomyces to much more far away species. You can see from Ascomycota to Basidiomycota. So you can see a trend. The color means this gene already identified to relate to ion responsive regulation. So you can see there's a trend from here to here. Type 1 system, the orthologous here, and their functions looks like more ancient, right? Because they cross different Ascomycota to Basidiomycota. And then looks like they evolved from this type 1 to type 2 because one more additional factor getting in and then evolved to a completely different type 3. So this is a kind of trajectory we can observe by just simply sequence comparison. And you can also see one more thing is here. This transcription activator doesn't exist in some faraway species. So you know this is a relatively new gene. So compare the emergence of this gene and their possible functional divergence across different species. We think this is a good candidate of microevolution in the evolution of transcription regulator network. So we will focus on this because KW is what we know before me. Okay, not my study. So we want to study this function in different species, these two other yeast species, and to see what will happen. Okay? And then, how to do it? Very simple. Actually, very simple. We want to know whether there is a transcription rewiring happen between these two species compared with the reference species. Very easy. We want to know their function. So we can compare the wild type with mutant to see whether they are required for ion responsive growth. And we can compare transcriptome because they are transcription factor, right? So we want to know whether they can compare wild type with mutant. We want to know what's the target gene. And by using a chromatin immunoprecipitation experiment, we can know the true target of this transcription factor. So we can compare all the information from here of these two species with the reference species. We will know whether they are the same or different. And then we can confirm this is a transcription rewiring or not. So by doing this experiment, very straightforward. We just use the condition to create high iron to low iron and knock out this gene. So this is the reference species. We know that the SEP1 is important, and delete it will cause it has a growth defect on the low iron condition. And then this is the out group. We will use the control here, and then we focus on these two species. So you can see Saccharomyces cerevisiae. Because we already know that actually is AFT1, AFT factor is required for this induction of ion uptake gene. So you knock out it will have growth defect. But in Latent screwberry is also the same, right? So we know this is what we see here, but what we are interested in is the SEP1. So you can see SEP1 here, they behave similar, and SEP1, knock out it, there's no growth defect, no growth defect. So which means SEP1, unlike Kendabican, SEP1 is not required for probably not required for ion responsive regulation in the turnover gene expression. Okay, so then we go to test a different condition and using chip to identify their true target. So we find out that in latency curvary, LK means latency curvary, the SEP1 actually they shift their target gene to different group of gene. So you can see this is Kendabican SEP1. We already know that SEP1 regulate multiple ion uptake genes, but it's also located in latency curvary. Only very few promoted targeted by SEP1, and only one is really regulated. So you can see it's an obvious difference. But what's the function of LK SEP1? So you can see we find out another group of genes. This is very small, but actually that belongs to TCA cycle. And you will find another latent group of SEP1 shifted to target to almost all of the TCA cycle gene and responsible for expression for about 30% of them. So once you test, you can see there's an opposite pattern, right? Kendabican SEP1 bind to ion uptake gene more, and latent group SEP1 target to TCA cycle gene more. So this is the opposite pattern of target gene and regular handover is a very typical feature of transcription rewiring. And to test its effect, we study the function of latent group SEP1. Because they target TCA cycle gene, we know TCA cycle gene is required for mitochondrial function, and mitochondrial function is required for growth on the respiratory condition. So we just use a simple growth assay to test that when we delete SEP1 in latent curvary, this yeast cannot have the respiratory growth. So we know that yeast can fermentate. So they will use glucose, D means glucose. They use glucose to fermentate to generate ethanol. And after glucose is consumed, they will try to use ethanol to perform respiration. So this growth stage means respiratory growth. So you see, in wild type, we add a respiration inhibitor. We inhibit respiration, so there's no respiratory growth. But deletion of SEP1 have the same effect. So we know that the SEP1 in latent curves is required for respiratory growth. So similar idea, we compete the deletion strain with wild type under the respiratory medium. So you can see the mutant has less competitive. And also we use another enzymatic assay to test the mitochondrion, one of the mitochondrial enzyme, and the SEF1 deletion has weaker mitochondrial enzyme activity, which means all these data suggest that the latent group SEF1 is required for respiratory growth and respiration. So which means in this species, they shift from, unlike Candabicum, they shift from iron to respiration. Okay? So it's very typical transcription rewriting case. And we go to another species, Saccharomyces cerevisiae. So we use completely the same strategy. Okay, we identify, oh, this is the chip binding target. You can see this color label with blue, very small, but the label with blue belongs to TCA cycle gene. So which means Saccharomyces cerevisiae SEF1 also bind to TCA cycle gene. And using geoanalysis, we know that, okay, the major target of this binding target belongs to TCA cycle gene and respiration. So based on the chip assay, looks like Saccharomyces cerevisiae SEF1 also target to TCA cycle gene, right? So looks like Lattice Groupery and Saccharomyces cerevisiae behave similar, but actually in the phenotype it's not. For example, we quantitate the gene expression. So these are all TCA cycle gene, and the blue color means they bind by the Saccharomyces cerevisiae SEF1. But if we compare the gene expression using microarray, we compare the mutant with the wild type. You can see delete of this gene will not cause majority of the TCA cycle gene misregulation. So this is very interesting. This transcription factor in Saccharomyces cerevisiae bind to a promoter, but remove it will not affect gene expression. Why? It's very special case, which means the phenotype may not decided by the misregulation of this TCA cycle gene, right? If we do the phenotypic assay comparing wild type and mutant. So here we also do another double confirm is that we fuse BP16 with SEF1 to make the SEF1 mutant become hyperactive to see whether the binding to the promoter still have effect. So if we compare this hyperactive SEF1 with wild type, we can find that this target gene got upregulated by the hyperactive SEF1. So which means the SEF1 binding to the promoters really has its function, but in the wild type, it has no enough activity to drive the expression. So this data just tell you that in Saccharomyces cerevisiae SEF1 is very strange. They see that on the promoter, but it doesn't responsible for driving the gene expression. Okay, it's different from Lattice Groupery, although the chip data suggests they regulate TCA cycle gene. Okay. So in the end, we want to test this phenotype again. So again, I told you that the SEF1 regulate TCA cycle gene and regulate mitochondrial function, so they will have respiratory heat tolerance phenotype. So using this phenotype, we can measure SEF1 is really required for this function or not. So we compare Lattice Groupery and the Saccharomyces cerevisiae by wild type and deletion strain and hyperactive strain. So you will find that as expected previously, Lattice Groupery SEF1, we deleted it, they have show growth defect. But in Saccharomyces cerevisiae, we deleted, we have complete opposite. Lattice Groupery SEF1 deletion has growth defect, but in Saccharomyces cerevisiae, it increased the bitterness. And in the hyperactive SEF1 will make cell grow even worse. So the phenotype in Saccharomyces cerevisiae cannot be explained by the SEF1 regulate this gene. It must be something else, right? So what is that? That's the interesting question we want to address. So probably because hyperactive SEF1 in Saccharomyces cerevisiae makes cell grow even worse, so we assume that there's a target gene that is deleterious, probably responsible for this function, because if we compare the SEF1 deletion wild type and hyperactive SEF1, this is SEF1 activity from 0 to normal to hyperactive, right? And they reflect the regulation. Support should be low, normal, and high. And we see the phenotype, it should be high bitterness to normal to low bitterness. So what's responsible for this must be a deleterious gene. So we use a lot of strategy to filter to find out the target gene from supplementary cerevisiae SEF1. So there are different criteria. I will skip the detail. But the final criteria we are using is we find out one of the most abundant target gene. So we decide to test it first. So this gene is called ND1. So simply the idea is if we assume ND1 is a deleterious gene, so we remove this deleterious gene, so suppose we can restore the phenotype. So that's what we did. So compared with the hyperactive SEF1 strain, we remove this gene, we find out that their fitness got restored. So this data suggests that actually is this deleterious gene responsible for this completely different phenotype in supplementary cerevisiae. Asians cruberry, the phenotype is explained by TCA cycle gene regulation. And in supplementary cerevisiae, although SEF1 also regulate, also bind to the promoter TCA gene, but they cannot decide the phenotype. They decide by another gene with stronger effect. Okay, so that's a spatial phenotypic outcome between these two species. And so, next question is we want to know how does ND1 cause deleterious effect? So previously ND1 already know is NADH dehydrogenase responsible for respiration pathway. And we also know that when this gene got super highly overexpressed, they will cause apoptosis. But however, these two situations cannot meet in our strengths because our strength will not related respiration, right? And also, the upregulation of this gene in our strain is not that high, it's not too overexpressed. So we are thinking about a third possibility. So we have another study that we use experimental evolution to study the protein formation. So yeast is a very special strain. The yeast species, they can live without mitochondria. Okay, so also they have a machinery try to maintain the mitochondrial stability. Okay, so this is a topical related to aging. But in fact, we use the ETBR to treat cell and let the cell evolve under this condition. ETBR will force the mitochondrial DNA to become unstable. So we evolve the cell in this condition. In the end, we will get the evolved clone carry different mutation that will have higher mitochondrial stability. So by sequencing this evolved clone, we find out that one of the gene have mutation on ND1. So we know that ND1 has another function. Also, okay, so this is the assay we are using. Now we use protein formation treated with ETBR. So we will find out there will be in a wild type there will be a protein formation rate, which means they lose mitochondria. But we find out that will make ND1 become loss of function. And another gene called HRD3, please remember this gene, we will mention it later. So this gene, when we become loss of function, they will make the mitochondrial DNA become more stable. So this assay just tells us that ND1 has a function to decide the protein formation, so decide the mitochondrial DNA stability. So we are thinking that maybe this effect is the deleterious effect controlled by ND1, so we want to test it. So very simple, we try to connect our SEF1 mutant with the protein formation frequency. So we will find out that when we compare with the wild type, deletion of SEF1 make the decrease the protein formation rate, and hyperactive SEF1 make the protein formation higher. But when we remove the ND1, we can restore this protein formation rate. So it looks like the SEF1 activity is correlated with protein formation. So we are thinking that the protein, this is a sick yeast cell, will decide the fitness and responsible for the defective growth. And the ND1 is the target gene with different level in wild type mutant and hyperactive SEF1 to decide this phenotype. Okay? So we use another evidence to support it. Remember previously I said HRD3 loss of function can also improve mitochondrial stability. So we try to put this mutant together with the hyperactive SEF1, try to see whether it can restore the phenotype, restore the fitness, and actually it can partially restore the fitness. And if we put ND1 deletion and HRD3 together, there's no even more improvement. So which means HRD3 is a kind of regulator, can improve mitochondrial stability, but upstream to ND1. So they have a hierarchical relationship. But this experiment just tell you that they can support that the SAFE1 control the phenotype in Saccharomyces cerevisiae actually decided by protein formation, decided by mitochondrial DNA stability. So this is not directly related to TCA cycle generation. So unlike Kindabicin, unlike latency curvary, SAFE1 in Saccharomyces cerevisiae have different phenotypic outcome decided by different set of genes, although their major triple binding target of SAFE1 is still TCA cycle. So it's a special case of transcription rewiring. They have maintained a major binding target, but the phenotypic outcome is opposite. Okay? So give you a simple summary here. So we identify this disruptive rewiring. If we compare reference species with these two clades, they will shift from ion responsive gene to TCA cycle gene. But between these two species, the phenotypic outcome is opposite because although they share the target gene, but in another species, the phenotype is dominated by another gene in this special species. So this is a kind of pseudo rewiring if we compare this species with this species. So we characterize two types of microevolution in transcription rewiring system. So one is disruptive rewiring, compare Kindabicin with these two, and another is pseudo rewiring if we compare these two species. And this is a kind of very small evogram in the transcription regulatory system. We use SAFE1 as an example. Okay? So then we go to the research interest 2 is we want to study the microevolution in the perturbed transcription regulatory network. So we use SAFE1 as an example. We know that in latent sclerosis, SAFE1 control multiple TCA cycle gene, right? So we detected we will find a lot of genes got dysregulated. They have fitness problem. So what kind of evolution it will happen if we try to evolve and select this mutant? Okay, so that's what I will do. So the idea is we already know the phenotype, right? From SAFE1 to these two phenotypes. So under this condition, we know they have growth defect, this already mentioned previously, but in the end, if we prolong the growth for many, many days, we will find that they will develop several fetal colony. So their fetal colony is much more better than the founder cell, the ancestral cell. So we assume that these cells will carry different mutation that allow the cell to survive under this defective growth condition. Okay, so this is a very, very fast evolution happening in the lab. They will develop a new mutation, and what usually we will assume these are suppressive mutation and suppress the previous defect. So actually, we collect many of them. You remember I collect more than 200 cells, and I try to evaluate their phenotype. So this is just an example. We find that there are two types. One, compared with the founder cell, we find that there's a type 1 evolved under normal temperature. They will always get better fitness compared with the founder cell. So this we call double compensation. This is type 1. Compensation. Another type is the cell evolved from higher temperature. We find that they will get compensation under the respiratory condition, but the trade-off means they got one food but another bad. Okay, so the trade-off they got defect in another condition. So we call this type 2. And we found out if we screen more than 200 clone, almost 90% of them show these two consistent phenotypes. So this is very strange, but the interesting case is that their evolution trajectory looks quite consistent. Okay. And this happened in the lab. And then by sequence, we check many clones. By sequence, then to compare, using whole genome sequence to identify the mutation. And then we compare the mutation hit the same gene. So these are different lines and different strands, and the mutation hit the same gene. So in the end, we using Venn diagram to compare them, and we find out that there are two genes hit by all of the strands. And in the type 2, there's another set of genes hit by all of the strands. And only this gene carry loss of function mutation, means sense and stop gap, which means loss of function. And this gene called friendship, usually we assume friendship mutation is loss of function. So both of them, they carry loss of function. So we think they are the potential causal mutation to cause the suppressive phenotype. And then to verify it, we just simply delete this gene in the SEF1 deletion strain or wild-type strain, and we can simply find out that by deletion IRA1, we can have the double compensation phenotype. By deletion AZF1, we can have trade-off and the compensation, so which means type 1 and the type 2 phenotype. So we can rebuild this phenotype. Okay, so this suggests that these two genes are the real causal gene. So this is a very lucky case because usually it's not easy to find out causal mutation. But in my case, looks like it's easier and simpler than I expected. And in the end, IRA1 is a kind of negative regulator to RAS PK signaling pathway. So actually, probably a lot of you already heard about this gene and this mutant because this happened in a lot of cancer cells. So looks like it's a general beneficial mutation happening in everywhere, not only yeast, probably also in mammalian cell system or mouse or whatever cancer, different kind of cancer cell. So that's why we will put more attention to AZ1 because this is a transcription, another transcription factor belongs to zinc finger transcription activator, and the function is unknown. And then we try to choose this for the further analysis. And then, so because it's a transcription factor, right? So we try to compare the knockout strain with the wild type to see what kind of gene got misregulated or changed in order to explain the phenotypic outcome. So we have found out, we know that these are belongs to type 2 phenotype of trade-off and compensation. So we use the gene ontology to study the RNA profiling, and then we find out under the fermentative condition, there are 4 categories of gene: carbohydrate, amino acid, and the negative regulator of RAS PK pathway, and the cell integral gene got downregulated. And under respiratory condition, they have anti-stress responsive gene got upregulated, and the translation-related gene got downregulated. And also we see an expected one, because SEF1 in Nations Coolbury got misregulated, downregulated, right? We deleted SEF1. But in the suppressive mutant, the TCS gene got restored back. And also more gene-related utilization, non-fermentable common cell got upregulated. So we want to know whether this probably can explain YBD trade-off phenotype and whether this gene can explain YB glycol compensation phenotype. So we use some phenotypic assay to verify the effect of gene expression. So for this unregulated gene, first the carbohydrate, we use the glycolysis inhibitor, and we find out that the delete of AZF1 will make the cell more sensitive glycolysis inhibitor, so which means this gene really matters. And then we use the nitrogen starvation to pre-culture the cell, and we find out that the deletion of AZF1 will make the lag phase become longer. So also which means the nitrogen and amino acid downregulation of this gene matters. And then we combine another synthetic loss of function mutation with this negative regulator, and we find out that there's a synthetic defect between the AZF1 and the knockout of this negative regulator. We find out that it grows even worse. Okay, so this synthetic sequence will suggest that this negative downregulation of this negative regulator also matters. And the final one is the cell wall integrity. We use Zymeworks treatment to remove cell wall. So if the cell wall is sick, so we use Zymeworks, the cell will more sensitive to cell wall removal, right? So we use Zymeworks to remove cell wall, and then we find out that the deletion of AZ05 with after AD detergent, the cell will lyse quicker and more effective. So also means all of this gene downregulation can respond for specific defect. So probably this phenotype and gene regulation can explain the YBD trade-off phenotype. And another one is we find out that anti-stress gene got upregulated under the fermentative condition, and the translation got shut down. So this kind of pattern, probably you guys also have some knowledge about that. When cells suffered in stress, they will turn on the protective stress, right? And they will shut down the translation in order to save some energy. So usually this up and down is quite common when cell encounter bad condition, okay? And one of the important anti-stress responsive genes is a lot of heat shock, chaperone, right? And one of the important transcription factor conserved from yeast to higher eukaryote like humans is HSF1. So we create a decrease of function mutant because this is an essential gene. So we only can use the decrease of function gene and combine with the AZF1. We find out that the AZF1 YP grass compensation phenotype will be destroyed if we put those decrease of function HSF1 into it. So which means that this anti-stress response relies on this quite conserved Hsiu transcription factor. Okay? And this we already know that because we evolved the cell under these two conditions. So that's the original purpose. We want to know that they should restore the gene expression. So we try to know that whether the restore of the TCA cycle gene is more specific to SEF1 deletion background, because we want to know this is a suppressive mutation or not, right? Because suppressive mutation, the definition is the suppressive mechanism should be more specific to the founder cell genetic background. So our founder cell is the SEF1 deletion background. So we compare the AZF1 deletion with the wild type and the AZF1 deletion under the SEF1 deletion condition. So you can see the restore expression of this TCA cycle gene compared with the orange and the blue color is higher. Okay, so compared with these two patterns, you can find the majority of them have higher aesthetic effect in the SEF1 deletion background. So which means this suppressive mechanism is more specific to SEF1 deletion background. So this is a very typical, quick, adaptive microevolution happen in yeast in response to dealing with a loss of function transcription regulating network in SEF1. Okay? So, very simple conclusion is we can evolve to let this microevolution happen directly in order to allow the cell to regain the fitness in a simple way and to compensate the misregulated gene expression. So this is a strategy for an organism that have bad mutation in the beginning but still can survive. So this is a very typical experimental evolution happening in the lab. Okay? And the conclusion of take-home message here is we use the evolution strategy. We have two evolution branch. One get the typical beneficial mutation, another get a specialized mutation. And then the idea is that if one transcription regulating network got abolished, actually the cell can evolve in another transcription regulating network in order to have a crosstalk and then try to restore the misregulated gene. And during this process, probably they will have a lot of side effects, so that's why we see some trade-off, right? So in the end, we know that there's a second perturbation in the modified transcription factor is a kind of mechanism that can allow the evolution of a new transcription regulating network without the original transcription factor. So this is a kind of artificial evolution, but we can see it in the lab. Okay, you know, evolution is the most difficult part is to see to believe. Okay, so this is a typical case of to see to believe. So the final part is very short because this is a very, very immature project right now, but I just want to share with you the idea. So as we mentioned previously, yeast is a kind of species who can live without mitochondria. But they still want to have mitochondria because mitochondria can provide them better fitness. We know a lot of function in mitochondria, right? So we use the experimental evolution, try to challenge the cell with ethidium bromide, which means can force the cell to create mutation in mitochondrial DNA to lose mitochondria. But we select it so only the fitter cell can survive under this condition. So that's why we use this kind of mutagenesis plus selective strategy. We can try to have evolved clones survive, carry mutation that improve the mitochondrial DNA quality control. So that's the strategy what we are using. We use mutation accumulation, we use selection, we got evolved phenotype, and in the end the purpose is we use forward genetics to try to understand the molecular basis behind this evolved phenotype. So here is the improved mitochondrial DNA quality control. Okay? So that's the idea, that's the result we have so far is using this strategy, we identify after 40 cycle of evolution, we identify many mutations. One of the mutations, as I mentioned previously, is NDU1 and HRD3. But actually, we try to test the more gene because we have a mutation spectrum, so we have a lot of candidates. So we find out at least so far more than 10 genes belongs to 3 completely different pathways. One is related to respiration, one is related to ER-associated degradation pathway, and one is associated with the proteasome, the protein degradation pathway. So knowing these 3 pathways and the target gene, we can extend the candidate by using the information of protein-protein interaction and genetic interaction. So ESA is a very convenient model organism because we have relatively comprehensive but clear protein-protein interaction map and the genetic interaction map. So we can expect once we get it, one gene, we can know who is intact with it, no matter it's physical or genetically. So by this way, we can have even more than 100 candidate genes. Probably their mutation can try to improve the mitochondrial DNA quality control. So now we are working on verifying these candidate genes. So we already have more than 20 candidates can confidently improve the mitochondrial DNA quality control. And next phase, we want to know why. And right now, the hypothesis is this gene can be explained related to proteostasis. And so far, based on I searched many literature, we didn't see very clear known hypothesis or model related to direct proteostasis from ER to control mitochondrial DNA quality. Okay, so I think this is a very there a lot of work can be done in the future, and I will do some genetic and probably because it belongs to proteostasis, right? So I will also focus on some of the proteomic analysis in order to know what kind of proteostatic state change happen in the mutant of this gene, and to try to build the molecular mechanism that how to improve mitochondrial DNA quality control. Okay, so I just combine all of my publication into these 3 interests. So, but I still a lot of details. So, if you are interested, you can take a look at my paper, and if you are interested to discuss with me. So, I think that's all today. I will happy to take some questions.
So I do have a question, not quite sure whether it's really naive. So it sounds like for the network to be able to rewire, it feels like for every particular transcription factors, it requires to recognize many different sites, right? So only a fraction of those are really functional, really regulate the expression of those genes. I'm just curious, so is that common for every single type of transcription factors, or the fraction of those functional fractions kind of correlate to the evolution age of that particular transcription factor? So maybe another way is to say whether during evolution the fractional functional site are going to increase or is going to maintain as a steady state.
The first question, I think this phenomenon that transcription factor bind to the promoter, but not important to the gene to drive the gene, is quite common. I try to collect several literature about they have both chip data and both the RNA expression data, and we find out that I call this non-functional binding, which means it binds to the promoter but not responsible for gene expression. And I find out that the percentage of non-functional binding can change from like several percent to 90%. Safe one is a very funny case. Actually, its binding target is more than 600 to 700. But it only responsible for expression for only like less than 5% of the target gene. But another case, if I remember correctly, HSF1, a Hsiu transcription factor, its functional binding is higher than 80%, which means it binds, it regulates. Okay, so it depends on different transcription factors. It can be very diverse. So I think it's a common phenomenon, but the percentage between functional and non-functional factors really quite diverse, depending on different situations. And one more thing, probably you can partially answer your second question is, I feel, that's how I feel. We know that there are some gene family in transcription, DNA binding domain, right? So zinc finger, that's a lot of big family. So their binding motif actually has some similarity. For example, CGG repeat. But the CGG repeat with some inter-motif sequence will decide specificity. I'm thinking that probably sometimes when the cell lose one zinc finger protein, probably they have another zinc finger family protein TF can replace that binding site, but with different affinity. Yeah, so I think that's one part of evolution mechanism, but I have no evidence to prove it. So I'm still thinking a project whether we can try this kind of phenomena, to test this phenomena in the lab. But yeah, but I need to have a very brainstorm for that. Yeah.
Okay.
So I have a follow-up question. So I thought before this talk, I thought transcription factor is very specific. So it's always a function. So you say that could be up to 80% is like non-functional binding. Is that just for the
80% is functional?
Yeah.
But again, some of them can be lower to.
Right. Is that just for the yeast, or do you see that the similar in human cells, or?
Okay. So the literature I collect from yeast, but I believe in other organisms they have the same situation. Because this is just a simple chip and the RNA-seq data, right? So you can easily compare. Yeah, so I think binding is always much higher than direct regulation. That's a common phenomenon, even in other organisms. Because it's not depends on organism, right? It's just the protein bind DNA sequence. Yeah.
So one of your data showing that SIP1 kind of lost it, or is regulate 4 different categories of the function. You have like the function of the TCA cycle, or do all these link together? Can you kind of link these 4 functions up, like there's a or even? So it's like they're totally right. Or I'm not talking about the evolution-wise, but just in that particular yeast, do all these functions are kind of linked together to kind of regulate some, or be regulated by something else?
That's a good question, but so far, SIP1 is a transcription factor that has very little information. So we don't know its upstream, we don't know its cofactor, we don't know whether it has compartment. We even don't know what kind of, for example, post-translational modification to regulate it. So we know nothing. And so that's why there are a lot of things we can do, I can do, but I cannot answer like what's the mechanism for that. Yeah.
So as far as I know, like for transcription rewiring, like one of the possibilities, as you say, that maybe like a similar transcription factor may take place once one is deleted or mutated. But the other 2 possibilities, one is like the DNA binding domain of the original transcription factor is slightly mutated, therefore the recognition sequence may be changed slightly. And the third possibility, that is the promoter mutation on the promoter or enhancer of the target genes. So in your study, can you like distinguish how the rewiring happens because of the DNA binding, because of like loss of completely loss of a transcription factor, or like change of the DNA binding domain of the transcription factor, or the mutation on the target gene enhancer sequence?
I think this is a very good suggestion or question. honestly, what I can say is, when we try to really understand how evolve from this type to this type, there are only two ways. First is we use evolution in the lab to see it happen. Second is we need to have enough species with different kinds of difference. And this difference looks like there's a transition from one type, one subtype to another type, and then they are comparable, and we see it, and then we can make a story based on that. So that's what I am trying to do. I try to use as many yeast species as possible, because we have another genome sequence right now, right? So some of them can use that to do experiments, some of them cannot, but we can do some sequence proper comparison. And what I can say is, if we are lucky, we really see, there's A type, there's a B type, and there's an intermediate type, and they share feature for both sides. So probably we can use that to build a hypothetical model, this is the transition problem from here to here. But if you want to say, is this always doable? I must say, if we are lucky to see that, we can say that. But in step one, right now, there's still a gap, because I cannot really find out intermediate species or intermediate type. So I have a plan to have a P2 lab in mind, because I tried. Remember, one of the reference species is Candida albicans. Candida albicans is a pathogenic yeast. So from Candida albicans to typical yeast like a baker's yeast, there's a big jump. So that's why I try to compare multiple pathogenic yeast relative to Candida albicans to see I can see a subtype that can have a transition from comparable feature from small difference, and then I can put them side by side. So that's what I can do. I plan to do right now. Yeah. So I cannot guarantee that I can really answer that question.
So is it possible that you pull the Candida albicans step 1 and put it into the step 1 deletion? So I'm thinking about if you could clone the Candida albicans step 1 and express it in the step 1 deletion of Cerevisiae. And then perhaps you could do more chip assay to see whether they bind to the TCA genes or it's still bind to the Candida albicans target gene.
But you mean I replace the also from Candida albicans to other yeast or vice versa? Yeah, I tried to do this, but I didn't proceed to chip experiment because we first, when we do also the replacement, like step 1 from one species to another species, we will check the phenotype first. So if the phenotype, like the growth defect, for example, didn't restore, I would expect they cannot work well. Or for example, they cannot bind to some of the target gene properly and drive the expression. But this can be a lot of issue. For example, this transcription factor cannot express well. This transcription factor cannot bind to DM well, or this transcription factor cannot interact with other general transcription machinery well. So that's why it has no activity. It can be everything. So that's why I stopped there, because I do also the replacement. I find out the fitness didn't restore, so I know it doesn't really work. So I didn't do, for example, chip assay to see whether it can still bind to different old target or new target. But this strategy, I think, is valuable because we can use this strategy to ask a different question. For example, we put a different osolo, just like we put different mutant form of a transcription factor. So we can see the difference between different osolo, whether they can decide different binding affinity, different binding specificity, and then answer a different question related to the evolution of transcription factor. So that's two different directions.
Sorry, due to the time limit, I think you can ask that question later. Okay, so I think the last minute I will give the microphone to our Hsiu. 好，那就谢谢各位。